couple of logistics things. I'm hot here. One, we've got just a couple presentations, and then we're going to have an open discussion. A housekeeping thing, I've had a request that if there are people heading to LAX and we're interested in carpooling, if you could meet probably just out here by the lobby and go, yeah, I'm heading to LAX, I'd be happy to share rides. I'll leave that up to you, but there was a request for that. That'd be great. And after the presentations, I'd like to meet with my speakers up here for just a few minutes. I have a couple of logistics questions I want to check with you on about things. And I'll ask you the questions now so you can think about it. One, we've had multiple requests for the presentations. So before I say, yeah, people can have them, I want your permission. And if you want them converted to PDFs, we can avail them to the participants. Then be thinking about whether or not you'd have any interest in writing these talks up as mini reviews for a special issue of either JQ or Agronomy Journal. So put that in your head. You can think about it during the last couple talks here. All right, looks like everybody's kind of settled back in their seats now. Oh, one other logistical thing. If you filled out the form, and please do before you leave, because we'll find this very valuable as we're planning ahead, give it to Hero. Hero, you want to wave again? She's in the back. Yep, she's back there. She's the one running around with the crutch. And, you know, hop along, Hero. Sorry, Hero, I haven't picked on you yet today. But hand those to her, and we'll be happy to take a look at those. All right, back to logistics here. Multiple folks have mentioned GRACEnet, and I thought, well, maybe we should let people know what this is just a little bit. So I'm going to take a few minutes. You should have been handed a brochure on GRACEnet. It gives you the websites and some other information on there. GRACEnet is the Greenhouse Gas Reduction through Agricultural Carbon Enhancement Network, and we've been working together for over a decade. I've had a couple people say, yeah, this workshop's been great, help me think about things. Well, that's kind of how a lot of us got started, is there was this idea of looking at measuring nitrous oxide, and a decade ago, there was a lot fewer people doing this sort of research. So we got together and we met in Fort Collins and we did things like, how do you put these chambers together? And we literally went out and we practiced pulling samples out in the field, and we learned from Arvid Moser and Tim Parkins and a few others. And Rod was there. He was one of the few people in the room that had ever done any of this before. And we've been off and running since, and we realized pretty quickly we had to get some consensus and some commonality on how we were doing things. So at the end of a couple of years, we ended up with sites across the United States, anywhere from 30 to 90 scientists probably, depending at the time and coordination, and the dots on the map change around a little bit. But there's a bunch of us, and we've been trying to work together for a long time. So our initial goals were to identify and develop agricultural strategies. As I was listening to the various talks, one of the things that I kept hearing is that you need to have an objective, and your methods need to fit that. So therefore, we use some of these combined techniques. How can we enhance soil carbon? How can we reduce greenhouse gases? And how can we improve environmental quality? So those were some of our goals. We needed some common approaches. We were working across the country in very different landscapes, different cropping systems. We needed ways of looking at carbon. We've alluded to the fact that soil is a big part of our nitrous oxide measurements. We could have a whole nother workshop just on how you do measure bulk density. And I'm hearing some chuckles. Yeah, you know what I'm talking about. And this wide diversity of different things you need to measure. So just getting us all on the same page is really important. So we have a website. We have a place where we can go to. We have an electronic book of protocols. And this is similar to the GRA's now book of protocols, that they started there and some of the same authors and then expanded and improved. We have a common set of spreadsheets that we put together where we try to capture all this metadata that you need to try to make sense of how people did things. And I'm hearing going, oh, this method is better. And I'm thinking over the course of the 10 years that I've been doing this or longer, that the methods for calculating keep changing. Now, one of the advantages of having these collective data sets that are becoming publicly available, we can go back and refine those calculations. The raw numbers are still there. We have some numbers, we can go back and look at them. There's ways to go back and improve our measurements. This data portal is publicly downloadable. So if you're somebody who's never done this and you want to have some clue if the numbers that you're getting make any sense at all, at least there's some place you can go look at somebody else's data and go, oh, okay, I'm in this area, and maybe my numbers aren't the same and the precision might be different, but at least you might find out if you're in the same ballpark. Find somebody that you can share samples with. See if your techniques are working. At one point, we all got sent out the same data set of numbers and ran through the calculations and sent them back to Tim Parkins to see if we could come up with the same numbers. The math in this can be tricky. That was an interesting exercise. We didn't all come up with the same answer out of the same data set, so have somebody check your numbers. This group has been very productive. I'm really proud of them because I've been watching them grow for a long time. So there's lots of publications out there this data has been used for. If you're going to get into this as a workshop type thing, or you want to start looking at collaborations, and Kurt alluded to this, finding these common protocols, getting some planning together, communication is critical, whether it's in a room like this, and I heard the comment before about they don't speak the same language, you know, and even if you're all speaking English, you're still going, what are you talking about? So communicating, challenges, buy-in. You know, even when we're looking at this, I get a little concerned about, have I been anal enough? Have I been paying attention to all those details? How many students and undergraduates and different folks have come through my lab? And you're going along, and if you're new to this, you could have another couple-day session on messy data. Why is this number so wacky? Did somebody turn these around, or was my vial compromised, or you name it, it can go wrong. And we didn't even talk about finger pokes out of needles. That's a whole nother thing. So I'm really excited to see all this enthusiasm and this idea of how can we do this better? How can we, you know, because we have some really big issues as a planet to solve, and if we need to be concerned both about comparing treatments across fields and short-term things, but we still actually need to be really concerned about accuracy. I think one of the things I've learned today is that we're always wrong. It's just how wrong are we, and what kind of error are we willing to live with? And you know, how do we decide? What's the most important thing, the 10% around the calculation? And then we compound that with the 100% variability from point A to point B, and this ephemeral effect. And we talked a little bit, Philippe, you know a little bit about this winter measurements. I'm in Minnesota, I'm one of those insane people. I have gone out, crawled in the snowbank, measured samples, because I need my zeros, because if you only measure the episodes, you're overestimating, because most of the year it's boring, they're flat. And what do we do with those non-detects? How do you want to calculate that? How do you want to handle that? That's a whole nother discussion. So lots of issues, because we want to be able to compare information, whether it's our colleagues in Brazil or China, or I'm not even sure how many countries we have here today. I suspect a few. I mean, we need to be able to talk to each other. And being able to talk globally is going to segue in to Mark, who's going to come up, and he's going to talk a little bit about another network of folks that's on an international level. And if Mark, you want to come on down, I'll pull your slides up for you. Yeah, I forgot Mark. I didn't forget Mark. Mark is special. I've known Mark a long, long time. He used to have hair when I first met him. He's going to smack me pretty soon. I'll let him just take care of this. I'll be nice, Mark.
Sure you will. Right. So, so, you know, I've been measuring gas fluxes in North Dakota for over a decade, and so I'm getting prepared for the winter. It's part of the protocol. Yeah, so thank you, Jane, for the introduction, and I'm going to just briefly talk or expand upon sort of this whole network issue, but at a slightly different scale on an effort that's going on through the Global Research Alliance. And I like the way Rod started the day with asking some questions. So I've got two, okay? We'll kind of finish up the way we started. How many of you consider yourselves to be strictly field researchers? Do you want to go out, collect your data, write it up, get into a publication? No real interest in doing the modeling, but just want to do the field research. How many in here want to do that? Okay, good. All right. Now, those of you who raised your hands, how many of you would like to see your data used in a broader sense, maybe to improve and validate models, and maybe even to put together some recommendations that might, have some scientifically defensible policy that could improve, greenhouse gas balance from agriculture? How many of you would like to see that your data used for that purpose? Okay, good. Just about a one-to-one almost. So, all right. So I'll get around why I asked those two questions in a bit here, but first, we'll talk about MAGGnet, Managing Agricultural Greenhouse Gas Network. Where does this? Well, yeah. I guess I should. What am I going to talk about? Well, I'm going to talk about a number of acronyms, because I feel it's important to kind of go through a linear step through the Global Research Alliance and what it is, because it took me a month to figure out what I was supposed to do 3 years ago with this effort, because it's kind of complex, the structure was. And then talk a little bit about MAGGnet, where we are as a group, and some of the things that we've been doing and what we hope to be doing here in the near future. So this all begins with the Global Research Alliance. And I think some of you already know a little bit about this. Alan is heavily involved in this. Global Research Alliance came about in 2009 as a result of sort of an outcome from the United Nations Convention on Climate Change in Copenhagen. At that time, my understanding is that agriculture really wasn't considered to be part of the overall solution for addressing climate change issues, and that was a big concern for the agricultural community. And so there was a recommendation put forth to say, hey, let's get countries together to kind of get some baseline information so that we'll be ready, so that when agriculture is considered to be an important part of this whole issue, we'll have some information already generated. And so that's what gave rise to the Global Research Alliance. New Zealand, the Netherlands, United States were big players early on here, and essentially the thrust of this group was what they want to do is that they want to make more food, produce more food, but with less greenhouse gases. So we're reducing the footprint of agriculture. Now, structurally, the Global Research Alliance is made up of 5 groups. Right? 3 that kind of carry over the large sectors of agriculture. There's the livestock group. There's the Croplands group, which Alan leads also with a colleague from Brazil in Embrapa. And then there's the paddy rice group, which is led by Japan. And then there are 2 groups that cut across those 3 groups. There's a soil and a carbon and nitrogen group. They're very heavily involved in modeling. And then there's an inventory and monitoring group, which is headed up by the folks up in Canada. And they cut across all 3 groups. So we've gone to down one level. We've got 5 groups. Now, within the Croplands group, they split themselves up into components. And the first component being quantifying greenhouse gas emissions from cropland. There's a second group that's composed of individuals from the northern European countries focused on greenhouse gas emissions from peatlands. And then there's a group led by the folks up in France, on nitrous oxide emissions and soil carbon stocks. They're all involved in modeling for that component 3. Now another level. Within component 1, okay, there's a work plan. And this work plan was put together, I believe, just after the first meeting that the Croplands group had in 2011, right? 10, San Antonio, Texas. Here, Long Beach, okay, all right, so we come full circle to here. So with that work plan, there was, they wanted to develop some protocols, develop an international database, put together some recommendations for practices for minimizing greenhouse gases from cropland, and improve upon emission factors, and then create summary documents for decision makers. And then this is where MAGGnet comes in, and that's the international database. And MAGGnet standing for Managing Agricultural Greenhouse Gas Network. And yes, it is sort of a play on words, because MAGGnet, we're hoping that we attract people and not repel them, in terms of getting people to be involved. Little catchy acronym. What is it specifically? It represents a coordinated multinational approach for inventory and analysis of greenhouse gas mitigation research. And I guess it's right now it represents sort of a clearinghouse for collecting metadata from experimental sites across all these countries that are part of the Global Research Alliance, where greenhouse gas fluxes or soil carbon dynamics and nitrogen dynamics have been used. But the focus here is to have sites where they have published data. That's really, really key. This project has been going on for just shy of 3 years. In that period of time, we have done a couple of metadata requests to members of the Global Research Alliance. We've updated our spreadsheet template, and we've also written a grant to help support some of the work associated with this, as well as some of the networking costs in terms of attending meetings for the Global Research Alliance. What is it specifically? I mean, right now we collect data from an Excel spreadsheet. An Excel spreadsheet that initially looked a lot like the data entry template that the GRACEnet group developed. In fact, I went to Stephen Del Grosso in Fort Collins, and I said, it's like, I want to be able to use a similar type structure, but without quite the amount of detail. But the key here is to encourage people to share information about their sites, but don't ask for so much data that it becomes so overwhelming that they're not even going to want to be involved. And so the tabs in here are relatively straightforward. I mean, if you had a paper from the work that you've done, you could go through there and enter a lot of this information. And the goal is that for each experimental site, it should take no more than 15 minutes. And I've tested this myself within my own sites, lay out the papers in front of you, go through the sheets, and it's 10 to 15 minutes. I can get it all in. So it's not that big of a commitment. At least I don't think. Of course, on the other hand, it depends on the country. If they really get involved, like France, they have close to, I think, 80 sites now, maybe close to 100, I don't know, they just added some more. It becomes a really big deal to add all that information, and then when you ask for an update, it's not all that easy. It takes months, actually, to do those, so it's pros and cons there, but we try to make it easy. We have a tab that's general instructions, so people can easily follow it. We use color-coded worksheets, and we really make use of drop-down menus, so it's not, we're kind of standardizing the input that we collect. Where is the distribution of sites? Well, we have 302 experiments included right now. This has sort of had a nice stair-step pattern over the last 3 years. The first metadata call had about 120 sites across about 10 countries. Last year at this time, if you remember, a few of you in the audience, I think have followed this over the years, we were just over 200 sites. And now we're just over 300. And we just had, just this past October, Argentina and United Kingdom signed on, and then also France added a number of sites. So we're continuing to build upon this and getting a really nice, I think, a collection of different climatic regimes. We just have about every surface soil texture covered except for just plain silt. So we're in the right trajectory. And also, linked to GRACEnet, the fact that GRACEnet's already organized and they have a data portal, it's relatively easy for us to go into that portal and extract out the key metadata and then to place it in here. And now I think there's 34 GRACEnet sites, if I'm not mistaken. So we, not in the domain, but by January we should be able to include those sites. Okay, all right. We also have university sites here in the U.S. also involved. At Kansas State, Chuck Rice has added some information for us, so we're starting to build upon that. So it's sort of an open door. How about the where are these experiments? How many, I guess, where they are in terms of their status? Most of them are completed, and you'd expect that for studies that have been published, but there's a lot of long-term studies that are going on here too, that are continuing. Most of these studies, they're measuring carbon, nitrous oxide, measuring it at most among the three greenhouse gases that are common to agriculture. And we also have information on production, which is key, particularly for projects where you want to kind of look at trade-off analyses. Are we sacrificing yield in order to mitigate gas fluxes? So it's nice to be able to have that information to test those sorts of things. What types of factors that are being analyzed across these studies? Well, there's obviously, you might expect a big focus on, if you're interested in nitrous oxide flux, then there's going to be a pretty big emphasis on fertilizer rate, fertilizer type, different types of organic amendments to the soils, but also people are interested in looking at tillage, crop production, crop type. You'll notice the pasture and grazing there. I mean, even though this activity falls within the Croplands group, we did not exclude grassland sites because we found that as we were getting input, there were systems where they were more integrated. So they had grasslands, they had grazed cropland, and at that point, we just didn't want to leave anybody out, bring them all in. And that allowed us to actually get some really unique sites in both in Uruguay as well as Switzerland. So we're being inclusive. So this kind of gets back to my first two questions of things that have kind of how MAGGnet's been used recently. In February, I was approached by Jean-Francois Susanna in France with INRA, and he was conducting a model intercomparison exercise where they're comparing different models, but what they needed to know is that they wanted to get some, find out what sites are out there throughout the world that have certain characteristics. And so they asked me to do a few searches to find certain types of sites using the MAGGnet database, and it was great because then we could identify sites, potential cooperators, and then I could channel that information to them, and then they could follow up with a specific PI for the project, and they could decide whether or not they wanted to be involved in the project. So that kind of gets back to my question, is that if you have a modeler who's really interested in doing a particular type assessment, say in semi-arid wheat systems, well, we have enough metadata here with MAGGnet to be able to search that out, and then the modelers can follow up and contact the PI to decide whether they want to participate. So that's really, I think, the first use we've had in MAGGnet for within 2 years of it being initiated. The other thing is that the Paddy Rice Group, they have, it's a different type of production. There's drainage issues, there's different types of soils and so forth. What they wanted to do is wanted to take the template that we created, but adapt it for their particular production systems. And so they did that this summer, and they've been able to sign on 5 countries, 13 sites, and I imagine that's going to be able to continue to grow in a part of the world where MAGGnet right now doesn't have that big of a footprint. But perhaps the Paddy Rice Group will be able to expand it with their template. And then finally, with all this, sharing information, you have to have some sort of agreement. These are often very common. You got to know, the data providers and the data users need to sort of have sort of a binding agreement about, credit and sharing and so forth. So we've created a very simple 2-page agreement using sort of patterning it off some agreements that were developed in Italy, as well as with Peter Grace's group in Australia that really made for a very nice, simple, easy to understand 2-page agreement that we're going to post on the GR website later on this year. Okay, so I talked about the grant. We hope to be able to attract more grant funds to be able to do assessments on our own with cooperators. Here we're working with 7 different countries, cooperators from 7 different countries to look at response data and for sites within the MAGGnet network. And so it's going to be those sites where we have a full slate of greenhouse gas data, soil carbon data, as well as yield, so we can do some trade-off analyses. But more broadly, I think this is one of these things, as a field researcher, I've been trained over 20 years where you have these tasks and you get them done and then you move on to the next thing. This is one of these things that's never really potentially going to end. You're always sharing about what the project is. That's why I think I'm excited about the potential series of publications where we could letting more people know about this, but you're also at the same time learning, particularly from the model community, and how we can amend the data entry template in order to collect information that is most relevant for them. Right? And also engage others in the scientific community to be involved. So that's kind of like an invitation. So if you're interested, you can see me. So thank you all for listening, and I know this is going to go right into our discussion session. I don't know if want me to take questions now, but.
Mark, are you using your database to identify gaps, crops that are underrepresented, areas that are underrepresented, situations like that?
In terms of, we could do that, absolutely. And
oftentimes that information is really useful in grant applications where it's been, your website has identified that this particular situation is not well understood.
Yeah, and just, it's sort of anecdotal, but we had a situation very similar to that at a meeting in Warsaw in April, and there was a question about potato production. And what do we have available? And we found out that we don't have a lot of sites that are looking at potatoes, and so that it identified a need. Yeah, you could probably help with that, right?
Maybe I'll ask a different question. For me, I'm not in the program, but I'm very interested in the N2O emission research. And under my unit, I'm from San Joaquin Valley Agricultural Science Center, Water Management Research Unit. I think it's a great location, but we don't have a national program project associated with the greenhouse gas emissions. We're under NP 211. I guess my question is, I'm interested in, but I know if I'm involved here, into the GRACEnet work, and it will take some time and commitment, to associate that.
Yeah.
And I don't know what's the mechanism, or the people who are in this. I understand that this is a work group, right? And are there like a funding agency, funding sources we can apply or something? Oh, I see. Oh, yeah, that won't work. That means that we have to judge if we have the time, we can make the commitment or not, into the program. And that's my.
Yeah, your last phrase hit it right on the head, right?
Yeah.
Whether that's for GRACEnet or even for MAGGnet, it's.
Bring your own resources.
Yeah, you bring your own resources.
Okay,
right?
Thank you.
Yeah.
All right, according to our agenda, what we have scheduled right now is pretty much an open discussion forum. And which means you can bring up any topic that you would like in terms of where we're at. I'm going to put Dr. Denmead on the spot, if he doesn't mind, if we could get just a few of his thoughts, considering that he's watched this system for a long time. Perhaps you can give some reflections. Or at least say hello to everyone. Introduce yourself, please.
Oh, well, my name is Tom Denmead. I'm a graduate from Iowa State, although thoroughly Australian otherwise. For my PhD, I worked on sort of a very primitive, I suppose, chamber system, in as much as it involved growing corn plants out in the open in containers. And so we have a sort of an open atmosphere, but we can measure the gas production, in this case the vapour production, by looking at changes in soil water content within the container. That's got nothing to do with what I did later, but it's been a great money spinner in a way that it's the one. I wrote a paper on it with Bob Shaw, and it's received more citations than anything else I've written. So for all the PhD students, make sure you do a good a good thesis, and then you're in business.
So did you students catch what he said there?
Well, they were lucky days. Later than that, I've been in CSIRO, the government organisation for scientific and industrial research in Australia, for a long time, and working in a division initially of plant industry, then in environmental mechanics, and lately in land and water. Where am I now? I think a few things that I've been involved in currently, and one particularly is something that I've jotted down here for something that you might consider for a future workshop, and that would be linking models and measurement. We've been driven by the fact that, okay, you've got the modelers over here, you've got the guys that make the measurements over there, but usually the modelers take their data from published stuff. In our case, we've set out to ask the modelers what they need us to measure in order to make their models good, and we've tried to see how far we can go in sort of satisfying those needs. And I think it's been pretty successful. One more thing to say is that if you do have a meeting next year, it will be my 60th year with CSIRO, and might be a good time to go out, so on a good occasion.
Thanks, Tom. I appreciate that. Any other dialogue? Like I said, this is an open time. Any of the discussions, if you had a question you didn't get to answered before?
Thanks. I had a question. I wanted to follow on that topic of measurement and modeling, and I asked this from the perspective of someone that doesn't know very much about modeling. But what are what am I thinking? Exponential decay, for instance. I'm curious if that's a component in a lot of the process-based models that exist, and what's the landscape look like for not just using our data to inform models for sort of global large modeling efforts, but doing model-assisted measurement when we're trying to quantify certain systems and treatment comparisons instead of having to rely only on our smattering of data points, but using the model alongside our data.
Michelle, can I put you on the spot in regards to the exponential decay? Is he in here? Where did he go? Well, I'm guessing that I think the answer might be no, but does anybody else have a comment?
So my name is John Knight at Sydney University, and I've recently been asked to work with some measurement people on doing models, and I think the answer, as far as I know, is that there isn't a specific model that predicts that exponential decay, although exponential decay in those kind of circumstances is quite a common phenomenon. So, and in fact, that's something that I'm now going to work on in detail. Yeah. But from the point of view of a modeler, I would say it would be great if someone could measure like every hour or something, you know, the output in a chamber, so that you know, so modelers can get more detailed measurements to work with, purely to test models. Whereas, you know, measuring twice a week is not really enough to test the details of a model. So I know modelers always want more and more data to test their models on, but that's, you know, that's a very real phenomenon.
So when I'm measuring nitrous oxide emissions and when a lot of other people measure nitrous oxide emissions, it seems somewhat common, especially when emission rates are low, to observe negative emission rates, and there appears to be some controversy over whether those are real. And I was wondering if anyone had any input on that subject.
I'll take the card and I'll right back to you.
Probably when you talk about negative, first you have to go in and, well, and I think Tim Park and Rod had the paper on that, is look at your minimal detectable fluxes and think about, is it within the error margin of your measurement, or is it really large enough to be considered to be negative? I mean, we don't usually think about a lot of nitrous oxide uptake, although if you get complete denitrification, of course, you're getting nitrous oxide further consumed and further reduced to N2. But I think you just have to think about your minimum detectable flux. If you have like 2 grams per hectare per day negative, well, that's probably just within the error margin of your measurements, and so the measurement error, and so you just need to go through a procedure to look at what your minimum detectable flux is to see, is it a real negative or is it just essentially no different than zero?
I've seen some that are strangely large, like double or 3 times the detection limit.
Then,
And I don't know whether to just discard those data or not.
yeah, there's a lot of judgment calls in this, and I think, you know, you have to look back at the data. I think, is there a reason that maybe something was unusual about that chamber or whatever? But yeah, large negatives don't seem to be the norm, but we always find out there's different processes than what we expect.
Why not? Because we know denitrification occurs. I think we have to be careful that we just don't reject the negatives because we don't like them.
Can the negative fluxes be associated with the fact that we're actually enclosing, we're enclosing within a chamber, and so if there is to be nitrous oxide uptake or consumption, there was no chamber there, it would otherwise disperse into the atmosphere.
Certainly my reading of some of the literature is that under normal circumstances, when the soil is not anaerobic, that nitrous oxide will be produced deeper in the soil and consumed close to the surface. So I think that, you know, it is possible that it's produced in one place and consumed in another. And if you have a whole bunch of nitrous oxide accumulating in a chamber, maybe it could be consumed by part of the soil. I don't know.
Rod, and then over here, I'm going to ask that you wait for the mic because we are recording.
Temperature increases in the chamber can create expansion and it could be an artifact of that. So temperature dynamics inside your chamber, I'm not sure that's something that was talked about today, but we did talk about insulating chambers. That is a possible explanation. So that would be something to look at. But as far as just discounting it because it's negative, if you've looked at these other aspects, if it's, you know, beyond your detection limit, I wouldn't say, I wouldn't recommend to just discount it because it's negative.
Thanks, Rod.
Yeah, I think it was Bill Schlesinger who presented a meta-analysis at ESA last year on N2O uptake, and he said nobody should do this analysis, but somebody like me who's well-established and old and has time to waste on looking at tons of old data that, no grad student should spend time doing something like this. He felt convinced that there's, at least looking at this large set of data, there's probably something real happening there, but it's not, maybe ecologically significant. But he did mention, a lot of data sets, a lot of papers don't even mention what they did with their negatives and don't have any, and how he felt it's important that people publish that. Maybe not make it part of the analysis, but at least mention in their publication that they found these rates and move them off to the side and maybe make that some supplementary data. I work mostly on methane, and so we have simultaneous uptake and production frequently. And so when you have net production versus net consumption, you're really working with a double exponential distribution and two different mechanisms that are being affected by two different sets of physical and biological environmental parameters. So I think it makes sense to separate it from an analysis if you're most interested in denitrification and production and looking at that transformation or that aspect of the transformation rather than the consumption, because that's really a separate thing. But I think it's important to publish it.
My group has been doing 2 years monitoring of N2O emission in the field. My interest, I have not been involved with this group. My interest is about the nitrogen mass balance. The California nitrogen contamination groundwater is a statewide concern. With that, and then I have the ability to monitor N2O emission. And from last 2 years, I see posters, presentations, and include this group. I just realized that there's very few people monitor what's happening in the soil. And we measure so much emission flux, we do those, we correlate with the management practice. But I realized, I think just a couple presentations, I saw people monitor emission. For those people who can do the modeling, especially soil physics, and we did soil gas into concentration monitoring over time. The data look fantastic. It has a positive correlation with the flux chamber measured data. So I would suggest if whoever had that ability and probably look into that. If you know the concentration in the soil, how that changes, you have some kind of interface modeling, and that can use those data, soil properties, bulk density, and the diffusion coefficient, that they can do a certain kind of modeling approach that way. Just a suggestion.
Other comments, discussion points? I'll give the call one more time. I know it's been a long several days for those of us that have been here for the entire week. So you guys have hung in there really well. And I warned my co-organizers I was going to put them on the spot for some last minute observations, and we'll start with Hiro.
First, thank you all for attending. I think we had a productive session or workshop, and I think it's nice to see dialogue and comparison between different techniques. Maybe we missed some points this time. I would like you to turn in your evaluation sheet, please, because from this evaluation sheet, I could figure out what we can do in the future to advance what we are doing here. so that our future analysis will be better. From few evaluation sheet I received, it seems there is some interest connecting to microbiology and presence of microbiologists. So that's one thing. Also, meteorology, maybe another, and data analysis and statistical analysis and interpretation is another thing. Any other thoughts? One more is linking the data with our measurement with modeling. So any thoughts on those area? Any prospect for invited speaker? Please let me know who is you intend to invite. Let me know. It will be nice to know special expertise with those people who have expertise like today. And I like finally to thank the speakers. I think everybody did a great job. We also have special thank to Jane and Alan. They've been running around with the microphone and looking around to see who was raising their hand. Sorry, I couldn't run around this time. Hopefully next year I could be running around. So, yeah. With that, thank you and have a safe trip.
All right, and Michelle, you dreamed up this. I remember having the conversation a year ago with you, and again, thank you. So I'm going to let you have one of the last words. I get the very final last word.
Okay, so I can't say anything about you, I guess, then, right?
Oh, you can say otherwise.
I know, yeah, so I won't. Well, thanks. I just to repeat, it's great to have so many people come, and thanks for coming. In addition, we had a very high demand and had to turn people away. I don't know how many people we had to turn away, but I guess I would say if you're part of one of the communities of ASA, what I learned during this process is that if you go to one of their business meetings and you're the only one with an idea to do something, you're going to have to work on it, which is actually a great thing. This has been a great pleasure to work with the organizing committee, which includes Hiro and Jane, but also Alan, and to be able to talk to all the speakers. It's been a great experience for me to have an excuse to talk to people about data and the details of how it's all done. I would just thank the ASA communities and everybody else who helped support this. I couldn't be happier with how it all turned out. It seems like we address things on, we can't address everything. There's so many minutia, but we address things on I think some real good general talks to get people going, and then some more basic kind of theoretical talks, some new ideas and things to think about. To me, it's been great, and I really appreciate everybody who's been involved. And safe travels, and don't have anything to say about you, Jane, sorry. Other than thank you.
You're welcome. All right, well, let's give a round of applause for all of our speaker one more time, please. Now give yourselves a round of applause for hanging in there and having really good dialogue. Thank you very much. So I'm serious. Compliment yourselves. And with that, just a reminder, I do need to speak with the speakers briefly after this, and there were folks looking to carpool to the airport if necessary. And unless somebody has a burning issue they'd like to bring up, I'll bring us to adjournment. I have a burning issue. You had the mic. You just want the last word.
Amir Satapur, who had to leave, asked me to mention something, and that is for vial storage. He was recommended to put his vials upside down in water and store them that way. And he even gave me a picture if anybody wants to see what that looks like, which makes a lot of sense.
That was a good last word. With that, I officially adjourn this workshop, and thank you very much for coming, and safe travels to everyone.